The citric acid cycle is a bit like a give a penny, take a penny jar. Imagine that you had a line at the supermarket and everybody in the line was taking a penny each time through. As long as everybody also added a penny, well, then this would be fine. The line would run smoothly. However, if somebody were to go through the line and take a penny but not give a penny, well, then the whole line would stop. The next person would be out of luck. You know, one of those people who comes in and just gives a nickel. This is similar to what we see with the citric acid cycle. Each time around the citric acid cycle, we're going to lose 2 carbons as CO2 in our isocitrate dehydrogenase and our alpha-ketoglutarate dehydrogenase complex. You could think of this as kind of losing 2 pennies each time through. If you were to add in 2 pennies each time through, therefore, you could keep the cycle running continuously. And as you run it continuously, you're producing NADH, you're producing ATP or GTP, and you're producing FADH₂. The NADH and the FADH₂ can go through the electron transport chain and be used to drive the synthesis of ATP. So as long as you are adding in 2 carbons, then each cycle, you can remove 2 carbons each cycle through these oxidative processes that are also going to power the production of ATP. However, if you do not, the way that you can add these carbons into the cycle is through acetyl-CoA. And where can you get this acetyl-CoA? A variety of different sources. So one of them is the pyruvate that we could get from glycolysis. We could also get pyruvate from the lactate from fermentation. We could get it from alanine if we were to break down amino acids. We could also get it from things like fatty acids. So lots of ways in which we can get this acetyl-CoA. We can get these 2 pennies that we're then feeding into the cycle. The acetyl-CoA then joins up with oxaloacetate to give you citrate. You go from 2 carbons, 4 carbons to 6 carbons. You go around the cycle, you lose 2 CO2, and voila. You keep this cycle going again as long as you're adding in another acetyl-CoA. So give 2 pennies, take 2 pennies, all's good. However, what if you have one of those people come in and take pennies but not give pennies? Imagine that somebody, instead of going right around this cycle, they take out the alpha-ketoglutarate to make glutamate, or they take out the succinyl-CoA to make some fatty acids, or they take out the malate to make into oxaloacetate to make glucose. In these cases, we're taking carbons out of the cycle, and we're taking more carbons out of the cycle than we're putting into the cycle. If we were to take this alpha-ketoglutarate out, or we were to take the succinyl-CoA out, or we were to take the malate out, then we wouldn't be able to make the oxaloacetate through the cycle that we would then need to have the 2 carbons come in and join from the acetyl-CoA. Therefore, we need to have reactions that are actually going to add to this cycle at points other than acetyl-CoA. We need reactions that are going to add more than 2 carbons. We're going to need reactions that are going to add 4 carbons, 6 carbons, 5 carbons. We're going to need these reactions in order to keep this cycle going. These reactions, these people that are adding those nickels, these reactions that are adding these carbons, these extra carbons are what we call anaplerotic reactions. And so today, let's talk about the anaplerotic reactions, how we can get carbons into the cycle by transforming glutamate to alpha-ketoglutarate and things like this. So let's talk about how we can add to the cycle so that we can take away from the cycle and still make this cycle useful for the production of energy. That is, we can use it for both anabolic, so building molecules, and catabolic, breaking down molecule purposes. So we're breaking down those big Lego structures into smaller structures, which we can use to build other things, or we can use to break down further, I guess, like smelt or whatever, and make some energy from. But by having these little pieces, they're really useful for taking out and doing things with. But if we're taking things out and doing things with them, then we need to be putting more carbons into the cycle. As you might imagine, however, this is going to require a lot of regulation. And so we're going to talk about how the cycle is regulated, as well as things like those anaplerotic reactions. It's also important to note that the citric acid cycle as a cycle is happening in your mitochondria, more specifically in your mitochondrial matrix, with succinate dehydrogenase embedded in that inner mitochondrial membrane, because remember that succinate dehydrogenase is that complex 2 of your electron transport chain. And so it has the job of transferring the electrons that are passed off from succinate into the electron transport chain. And so it has to be embedded in this membrane because it's that flavin protein that needs to be part of this chain. So as a cycle, this is happening inside the mitochondrial matrix. However, some of these enzymes also have other roles, and not all of those other roles are going to be inside of the mitochondrial matrix. A great example of this is malate dehydrogenase. So yes, in the citric acid cycle, malate dehydrogenase catalyzes the conversion of malate into oxaloacetate. However, we also saw how malate dehydrogenase could play other roles. In the cytoplasm, it can reverse the directionality of this, and this is going to allow you to bring NADH out of the mitochondrial matrix, or NADH equivalents, because remember, the NADH itself cannot come in, which is why you're sneaking those electrons from oxaloacetate onto malates to bring them out, and then switching them back out onto oxaloacetate to then use them for gluconeogenesis. We also saw how we could kind of do things in the reverse direction, how we could use malate dehydrogenase to reduce oxaloacetate in the cytoplasm, and then reverse the situation inside of the matrix in order to bring reducing equivalents of NADH into the matrix, because we couldn't bring the NADH in. In these examples, we're seeing malate dehydrogenase play roles outside of its role in just directly in the cycle. We're seeing it in other cycles, like the malate aspartate cycle. And so this is just to demonstrate that we could have cytosolic and mitochondrial versions of some of these enzymes. We have different isoforms of some of these different enzymes. These enzymes don't have to function just as part of the cycle, but they are key components of the cycle. So just if you see one of these enzymes acting elsewhere somewhere, don't be surprised. You have a lot of copies of these enzymes, and they can kind of like hang out together to keep the cycle going. They can also hang out in other places and have other functions. And when I say other functions, it's kind of like other metabolic functions, but still catalyzing those same reactions. And sometimes, like with malate dehydrogenase, catalyzing these reversible reactions, where you can see it catalyze this reaction in both directions, depending on the situation. This is going to complicate things a bit when we think about regulation. When you think about the regulation of the citric acid cycle, I want you to focus on thinking about the regulation of the cycle, rather than thinking about the regulation of these enzymes and their other roles. Hopefully this will make more sense when we get into talking about the regulation, but if we think about the cycle, as a catabolic cycle, then you want to inhibit it when you have a lot of energy and make it go when you need some energy. Because when you do the cycle, you're getting the NADH, you're getting the FADH₂, which you can cash in for ATP, and you're getting that GTP or ATP, depending on the isoform, directly in the cycle itself. So we'll look and see how some of these steps are going to be highly regulated, and we're going to be discussing several of them in particular: citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. But that does not mean that these other steps don't have regulation, and sometimes this regulation is even more important for their other functions. And so the regulation, say, of malate dehydrogenase is going to be very important, but it's not considered one of the main regulatory points of this cycle in most cases. And therefore, I haven't highlighted it on this figure. There are a couple ways that we can regulate things. Well, one way is just regulating how much of various things we have. Just based on the concentrations, we can kind of be modulating whether the reaction is going to go or not, especially when we have steps that are going to be easily reversible. We can actually change what direction the pathways are going. We also can have enzymatic regulation. If we think about where we would want to have enzymatic regulation, that would be kind of like the steps in which we don't have that reversibility. Those steps where maybe we're losing CO2, we're using energy, steps like this, where we have these kind of like go, no go points, we can regulate these enzymatically. And in fact, if we look at the citric acid cycle, we'll see that there's going to be a few core enzymes that we're going to regulate enzymatically. This is going to be this pyruvate dehydrogenase step, which technically isn't part of the citric acid cycle, but it's really important for getting into the citric acid cycle, as well as the citrate synthase. So when we're combining oxaloacetate and acetyl-CoA, this isocitrate dehydrogenase step, where we are taking isocitrate and oxidatively decarboxylating it to give us alpha-ketoglutarate. And then this alpha-ketoglutarate dehydrogenase complex step, where we are basically oxidatively decarboxylating alpha-ketoglutarate to give us succinyl-CoA. If we look at these steps, well, what do they have in common? What they have in common is that these are kind of going to be steps where we are basically losing CO2, we're generating NADH. These are going to be kind of like energy generating steps because we're generating that NADH and we're releasing this CO2. So these are going to be like these highly energetic steps, steps that we can't easily go back from, steps that we're going to want to regulate heavily. So remember, we can have allosteric regulation, where we have small molecules bind and affect the activity of something. And then we can have covalent regulation, where we have things like phosphorylation that are altering the activity of the enzyme. If we look at what is allosterically regulating these enzymes, we can see that they have a lot of the same things that are regulating them. If we look at what's activating them, well, in the case of our pyruvate dehydrogenase complex, we have AMP, CoA, NAD+, calcium. As in this citrate synthase, we have ADP. Isocitrate dehydrogenase, we have calcium and ADP. And alpha-ketoglutarate, we have calcium. What do we notice if we kind of compare them? Well, we can see that they have shared regulators so that we can kind of regulate the pathway as a whole rather than just regulate one at a time. But they also have some regulators that are different, which allows us to have further regulation since we can do things where we can take molecules out at different points along the way. First, let's think about what these different regulators imply. Keep track of these different allosteric modulators in your metabolism spreadsheet. Follow along and do this for the citric acid cycle, and then go back and do it and look and see where you saw those same regulatory molecules in our pathways, like glycolysis, gluconeogenesis, glycogenolysis, glycogenesis, because we'll see that a lot of these same allosteric modulators are going to be used. Why is this? This gets to that point about like, what does its presence indicate? If we look at what is activating these, we see a lot of them are activated by AMP or ADP. These are going to be indicators of a low energy status. If we had a lot of ATP, that would be an indicator of a high energy status. We can see that these enzymes are going to be inhibited by ATP and activated by AMP or ADP. This makes sense because, well, this pathway, remember, if we're doing it in its entirety and we're just kind of like cycling around and around, what we're doing is we're generating a lot of our NADH and our FADH₂, as well as one direct GTP or ATP equivalent. So we're generating a lot of energy. If we have a lot of energy already, we're not going to want to keep breaking things down. Instead, we want to slow the TCA. And maybe then instead of going down in the TCA and breaking everything down, we can do all sorts of more anabolic stuff. So if we got a lot of energy buildup, we don't want to keep doing this. If we have a lot of AMP, we have low energy, then we do want to go through this whole pathway. And so that's what we're going to do. Similarly, this pathway generated NADH and it used NAD+. If we have a lot of NADH already, we're not going to want to do this pathway. And so if we have NADH, that's actually going to inhibit these enzymes. It's really important that we don't build up too much NADH. We saw when we talked about glycolysis, how we needed to regenerate that NAD+ if we wanted to keep things going. And that was why we needed to sometimes do things like fermentation to regenerate that NAD+. In addition to being needed to keep glycolysis going, it's actually going to also be needed in order to keep our citric acid cycle going, because we're going to need to have that NAD+ as one of the reactants for these steps. But the bigger problem with not having enough NAD+ is really that you have too much NADH. If you have too much NADH, well, NADH is actually going to inhibit these steps. And if NADH is inhibiting these steps, then you can't have the cycle keep going. Looking at the electron transport chain, you could have problems where electrons could escape and cause oxidative stress. We wouldn't want any of those things happening, so it's important that we have the cycle be regulated by the levels of NADH, and we don't have our NADH actually building up. If we have NADH built up, often what's going to happen is then we get our pyruvate converted to lactate with the help of lactate dehydrogenase. We regenerate that NAD+. That NAD+ is now going to stimulate our pyruvate dehydrogenase, and it's going to allow us to keep this cycle going. But when we had the NADH built up, then that's going to inhibit the pyruvate dehydrogenase, inhibit the citrate synthase, inhibit the alpha-ketoglutarate dehydrogenase complex. And we saw before how it was also going to inhibit our pyruvate kinase step so that we could stop glycolysis from making that pyruvate, which we can then take through the citric acid cycle. Let's see what else we have as allosteric regulators. One of them we'll see is calcium. I don't need to be too concerned about the calcium, but basically the concentration of calcium in your mitochondria where this is taking place is going to depend on the electron transport chain functioning. It's kind of like import into the mitochondria depends on that gradient. So if the gradient is functioning, if you've got that electron transport chain functioning, you're going to have the calcium in the mitochondria, and that calcium in the mitochondria is now going to stimulate this alpha-ketoglutarate dehydrogenase complex. It's going to stimulate this isocitrate dehydrogenase complex, and it's going to stimulate this pyruvate dehydrogenase complex. So calcium is serving as an indicator of the electron transport chain functioning. When it's functioning, then it's okay to keep things going with this pathway. But if it's not functioning, then you're not wanting to go down this pathway. Okay, what else? We have Acetyl-CoA is going to inhibit citrate synthase. Well, if we look at Acetyl-CoA, we can see that it's just a downstream product in this pathway. This is just an example of feedback inhibition, like we've seen before. Similarly, it's inhibited by citrate, another example of feedback inhibition. We'll also see Acetyl-CoA can inhibit alpha-ketoglutarate dehydrogenase complex. So voila, we get kind of like this mutual inhibition. But we can see that citrate synthase is inhibited by citrate, whereas when it makes sense for alpha-ketoglutarate dehydrogenase complex to be inhibited by citrate, because citrate is actually a precursor to making this alpha-ketoglutarate. You inhibit it when it's a product, but you would stimulate if like it was something upstream, but we're not going to stimulate, we're just not going to be affected by it. And so that is kind of like the core of this. We can also see with our pyruvate dehydrogenase, its product was acetyl-CoA. And it's going to be feedback inhibited by acetyl-CoA, just like we saw down here, but with a different molecule. So acetyl-CoA is going to inhibit pyruvate dehydrogenase. This is going to be really, really important because acetyl-CoA isn't just coming from pyruvate. In fact, one of the sources of acetyl-CoA is actually going to be fatty acids. And so if we're breaking down a lot of fatty acids, we're going to get a lot of acetyl-CoA. And if we get a lot of acetyl-CoA, then that's going to say, hey, pyruvate, stop breaking down and making more acetyl-CoA. We've got a bunch of acetyl-CoA already. So we want to stop making more. It's really important that we don't just keep making acetyl-CoA, because what can actually happen is that when you have a lot of acetyl-CoA, they can actually like join together to form these things called ketone bodies. And these ketone bodies can actually go through the bloodstream. And they're good for getting energy to your brain when you don't have glucose, but they're not so good if you have too much of it. And because what they can do is they can do things like actually acidify your bloodstream because they're these carboxylic acids. We've heard about like ketosis or diabetic ketoacidosis. That can happen because you have a buildup of acetyl-CoA. Similarly, we'll see that pyruvate dehydrogenase is going to be inhibited by fatty acids because if you've got a lot of fatty acids already, you're not going to want to make more acetyl-CoA because acetyl-CoA can be used to make fatty acids. So this is kind of another example of this feedback inhibition. The regulation of pyruvate dehydrogenase is more complicated than the other ones, in part because acetyl-CoA can be used for so many different things. The pyruvate dehydrogenase complex, not only is it regulated by those allosteric modulators, but it's also regulated by phosphorylation. There are enzymes called pyruvate dehydrogenase kinases that can phosphorylate and inactivate PDHC, and then phosphatases can activate it. So the kinase inactivates the complex. So the things that activate the kinase inactivate the complex, and the things that activate the complex are the things that inactivate the kinase. Don't worry about this, just know that it is regulated at this additional level. Whereas the other core regulatory points in the citric acid cycle are regulated basically just on these allosteric modulators and the amounts of different things regulating the other steps. When we talk about the regulation of the citric acid cycle, if you think about it for taking components out, then your brain gets a little scrambled. But the logic makes sense if you think about the pathway is making NADH and making ADP. If you have a lot of those, you want to inhibit the pathway. And if you don't have a lot of those, you want to stimulate the pathway. Similarly, if the electron transport chain is going, you want to keep this pathway going. If the electron transport chain is going, that's going to be indicated by the calcium. And so that's going to help stimulate the pathway. So in terms of the regulation, that's what I want you to focus on, is those core regulatory molecules, and then see how those molecules regulate other enzymes in other pathways and see whether you notice anything about what they might have in common or what they might have different. We talked about the regulation. Now we're going to be talking about what can we do with the parts and where can we get parts from. One of the things that we can do with the acetyl-CoA is we can make fatty acids and we can make those ketone bodies. We'll talk a lot more about this when we talk about fatty acid metabolism, but fatty acids are synthesized in this process from acetyl-CoA, where you basically join them together using this enzyme, fatty acid synthase, which is really cool, and we'll talk a lot more about it. The process is going to use NADPH, which remember was one of the reasons why we needed the pentose phosphate pathway, which generated NADPH. This process uses acetyl-CoA, and that's why the pyruvate dehydrogenase complex is also going to be regulated by fatty acids and by acetyl-CoA, because the pyruvate dehydrogenase complex is kind of going to connect that pyruvate to both the making of the fatty acids if you want to store the energy, or it can be linked to the breakdown if you want to break down the pyruvate further to get energy. One complicating factor is that, well, the fatty acid synthesis is taking place in your cytoplasm, but the breakdown through the tricarbocylic acid cycle, and even if you don't break down the whole thing, the pyruvate dehydrogenase step, which you need to make the acetyl-CoA, this is happening in your mitochondrial matrix. Remember that we had to take pyruvate into the mitochondria to make acetyl-CoA there, rather than make acetyl-CoA in the cytoplasm, because acetyl-CoA couldn't get into the mitochondrial matrix. Similarly to how it can't get in, it can't get out. So we can't take acetyl-CoA directly out. Instead, what we have to do is we have to take citrate out. We take pyruvate, we make acetyl-CoA. That acetyl-CoA can now be joined to oxaloacetate to give you citrate, and citrate can be taken out of the mitochondria. We then have an enzyme, ATP citrate lyase, that what it can do is it can basically take that citrate, take a CoA, take an ATP, and split the citrate into oxaloacetate and acetyl-CoA. Don't worry about this for now. Just know that I want you to know for now that citrate gets taken out of the mitochondria to make fatty acids and more ketone bodies. When we talked about gluconeogenesis, remember that's the making of glucose from our non-carbon sources like pyruvate. If we start with pyruvate, then in order to get to phosphoenolpyruvate, we had to make oxaloacetate as kind of like this intermediary. We use pyruvate carboxylase to make oxaloacetate, and then we use PEP carboxykinase in order to make our phosphoenolpyruvate. Remember that the pyruvate to oxaloacetate step, this pyruvate carboxylase step, this was happening in your mitochondria, and then the PEP carboxykinase step, that could happen in your mitochondria, or that could happen in your cytoplasm. We can see that in our citric acid cycle, we have oxaloacetate. And so like you could think, okay, well, why can't we just take this oxaloacetate and use it? If we were to say, look at our pathway again, couldn't we take that oxaloacetate and just use PEP carboxykinase to make phosphoenolpyruvate? And well, yeah, you kind of can. I say you kind of can because you can't take this oxaloacetate that you have in your mitochondria, but you can take oxaloacetate that you have in your cytoplasm. Because, well, what happens is oxaloacetate itself is one of those molecules that can't go in or out of the mitochondria. Similarly to how we couldn't get our acetyl-CoA through the in or out of the mitochondria, and how we couldn't get our NADH in or out of the mitochondria, we can't do that with our oxaloacetate. So instead, if we want to make glucose, we take out malate. When we take out malate, we can then convert it with malate dehydrogenase to give us oxaloacetate. We can then use that oxaloacetate that's in our cytoplasm. We could use our PEP carboxykinase in order to make phosphoenolpyruvate. Now we could go through the cycle of gluconeogenesis. When you take things out, the problem that comes is that then you need to find a way to keep the cycle going. With each turn of the cycle, we're losing 2 CO2, and we enter 2C as our acetyl-CoA. So we have 4 carbons in our oxaloacetate, 2 carbons in our acetyl-CoA. We join them together and we get the 6-carbon citrate. We lose 2 of those, and we're back to having a 4-carbon compound. We add 2, we go around, we lose 2, we have 4, we add 2, we lose 2, we add 2, we lose 2. We keep this going, and we're generating ATP. So as long as we keep adding acetyl-CoA and we don't take anything away, we can keep this going. Now imagine, however, that we took out the malate, say because we wanted to make glucose. In this case, we basically remove 4 carbons from the cycle. In order to put acetyl-CoA into the cycle again, we needed oxaloacetate. But we can't put acetyl-CoA into the cycle if we don't have oxaloacetate. And we won't have oxaloacetate if we took out malate because, well, malate was giving us oxaloacetate. So how do we get acetyl-CoA into the cycle? We can't, unless we had oxaloacetate present already. And so typically we already have oxaloacetate present, and it's not like we only have 1 molecule of each of these things. So we could put acetyl-CoA in and the cycle would go if in like normal conditions. But if you think about only having 1 molecule of each of these, well, now that wouldn't work. And so on net, you wouldn't be able to, say, make glucose if all you put in was acetyl-CoA, because you need to have more carbons in the cycle to keep things going. So if you had the cycle like fully stocked and you put in acetyl-CoA and you kept putting in acetyl-CoA, but you kept taking out malate, the cycle would stop because you wouldn't have enough carbons. You're putting in 2 carbons, but you're taking out more than 2 carbons, you're taking out 4 carbons. So you're taking out more pennies than you're putting in, and the cycle can't go. Now I'll imagine, however, that you put in something that was later on in the pathway. Instead of putting in just acetyl-CoA, say you put in citrate. Citrate, remember, this was going to be 6 carbons. So if we put in citrate, well, now we're good to go. We're adding in 6 carbons, but we're only taking out 4. And so voila, we can replenish this pathway. This is an example of what we call an anaplerotic component. For something to be anaplerotic, it must put in more carbons than get taken out. So you lose 2 carbons as CO2 per turn of the cycle. And so we need to add something greater than 2 carbons in order to keep it going. A lot of the time, we can also call these anaplerotic things, we call them glucogenic, because that means that we can basically make glucose from them, because we could take out the malate from the cycle and we could use that malate in order to make glucose. And so therefore, by putting in something like citrate, we're able to make malate and take out the malate, but we still had, because we put in 6 carbons and we only took out 4, we're still on net going to be able to keep this cycle going. So as long as we're putting in things that are more than 2 carbons, we're able to keep the cycle going. But if we're only putting in things that are 2 carbons, well, now the cycle can't keep going. And this is why things like our fats are not considered glucogenic. So we'll talk more about this when we talk about our fatty acid metabolism. But basically, when you break down some fatty acids, you get acetyl-CoA. We can put acetyl-CoA into the pathway, but only if we had oxaloacetate present already. If we didn't have oxaloacetate present, because say we're using malate to make glucose, then what would happen is we wouldn't be able to put acetyl-CoA into the pathway, on net at least, and so therefore we wouldn't be able to sustainably make glucose just from acetyl-CoA. Turns out that like bacteria and plants and some microbes, they have this cool like bypass called the glyoxylate shunt. We do not have this, but what they can do is they have these extra 2 enzymes, isocitrate lyase and malate synthase. This allows them to kind of like split isocitrate into this molecule called glyoxylate, as well as to a molecule of succinate. Succinate can enter the pathway. Glyoxylate can combine with the acetyl group of acetyl-CoA to give you malate. Malate can enter the cycle, and you can keep things going, even though you're only adding 2 carbons. And the reason why you're doing this is because it allows you to bypass those steps at which you were losing the carbons. You're also therefore bypassing the steps at which you were generating most of your energy. However, this is going to be used kind of like anabolically in order to keep making malate and oxaloacetate so that you can do things like keep making your glucose. So it's used more like anabolically. But again, this is not a pathway that we have. And so therefore, in order for something to be anaplerotic for us, we have to add things that are greater than 2 carbons. This distinction is easiest to see if we think about amino acid catabolism. We'll talk a lot more about amino acid catabolism, but the bottom line is that we can take amino acids and we can remove the amino group from them, and that'll give us an alpha keto acid. So basically, remember, an amino acid that had an amino group at the alpha position to that central carbon. Here we have a keto group, alpha to the central carbon, so we have an alpha keto acid. It's what we get if we basically remove the amino group from an amino acid and exchange it for a carbonyl. When we do this, well, now we have an alpha keto acid. And if we look at our tricarbocylic acid cycle, we can see that we actually have a number of alpha keto acids here. For example, if we take the amino acid glutamate and we remove its amino group, we get alpha ketoglutarate, which was a component of our citric acid cycle. Similarly, if we take an amino group from alanine, we get pyruvate, and if we take an amino group from aspartate, we get oxaloacetate. I don't need you to worry about these right now. We'll get much more into them in the future, but just know that basically we can take amino acids, we can remove the amino group, and we can get things that we can enter into the pathway. Rather than just removing the amino group, we'll see that we actually typically take the amino group and put it onto alpha ketoglutarate to give us glutamate. And then that glutamate is often then going to be what we have feeding into our citric acid cycle. We'll talk a lot more about this when we talk about the amino acid metabolism. For now, just know that the skeletons from the amino acid can enter the pathway. But depending on what amino acid you start with, you'll get different alpha keto acids left over. Some of these can directly enter the pathway, and some of them can be converted into the things that can enter the pathway. Some of these are going to be considered glucogenic, and some are going to be ketogenic. The ones that are glucogenic are things that are anaplerotic for the pathway. So remember, that means that they have to have more than 2 carbons because they have to be adding more to the cycle than they're taking out. You got to put in more pennies than you take out in order to keep this cycle going. However, if you add anything in this cycle that's basically further down than the acetyl-CoA, so if you're adding something that's already in the cycle rather than just the acetyl-CoA, well, now you can keep the cycle going. Some of these amino acids say they break down and give us pyruvate. If we look at pyruvate, here things get a little trickier because it looks like, well, it's outside of the pathway, so it wouldn't be anaplerotic, right? However, if we think about pyruvate, we can do 2 things with it. It can enter the pathway either as acetyl-CoA if we used our pyruvate dehydrogenase, or it could enter as oxaloacetate if we used our pyruvate carboxylase. If we enter as oxaloacetate, well, that was something that was in the pathway, so pyruvate is anaplerotic. It can be used to generate oxaloacetate, and therefore things that generate pyruvate are also going to basically be glucogenic. What about things that only gave us acetyl-CoA? So in that case, it would just be ketogenic. So this would be our leucine and our lysine. We call them ketogenic because they can be used to make acetyl-CoA, and we talked about how acetyl-CoA, if you have a lot of it built up, it can join together to make those ketone bodies. So those amino acids that are going to be ketogenic, we say that because they make acetyl-CoA or they make acetoacetyl-CoA, which can be used to make those ketone bodies or to make lipids. But they cannot sustainably be used to make glucose. This does not mean that if you took an amino acid that was, say, radioactively labeled and you fed it to the animal or whatever, you could find it in glucose. Don't be too surprised if you find it in glucose. But if you found it in glucose, it was because the cycle was already being able to keep going because you had components in there already. You wouldn't be able to sustainably make glucose because you would be putting in 2 carbons and taking out more than 2 carbons to make the glucose, and so the cycle would stop. We don't have that glyoxylate shunt, we wouldn't be able to do that. One of the clearest ways to kind of like illustrate the difference between ketogenic and glucogenic is if we look at the breakdown of branched chain amino acids. If we break down valine, what we end up getting is succinyl-CoA. If we think about succinyl-CoA and we think about our pathway, succinyl-CoA was in our pathway, so this would be glucogenic. And we weren't generating acetyl-CoA, so it wouldn't be ketogenic. That does not mean that we couldn't use it to make fats, however, because, well, then once this goes through the cycle, it'll keep going through the cycle, and then you can end up making acetyl-CoA, and you took that citrate out and made lipids. So these distinctions are kind of They're kind of talking about kind of like the first pass, like on at first net, but then like everything kind of gets jumbled up together. And so once you have the cycle go around kind of like once or things, all bets are lost. These molecules are interconverting and it gets complicated. But that's the basics. We would call those glucogenic. And that was our valine. It was making our acetyl-CoA. If we think about leucine, well, leucine, if we break it down, we get acetyl-CoA and acetyl-CoA, and those were going to be ketogenic. If we think about isoleucine, though, it's going to give us acetyl-CoA and acetyl-CoA, and so it's going to be both ketogenic and glucogenic. Again, we'll talk more about the actual catabolism of the amino acids later, but that's going to be kind of the bottom line is that when you break them down, you get skeletons that can enter at different points. If they're entering as things that are already in the cycle, we call them glucogenic. If they're entering as acetyl-CoA, we would call them ketogenic. Because if you're only making acetyl-CoA, you can't take components out to make glucose. Therefore, if you're only making acetyl-CoA, you wouldn't be glucogenic. You couldn't on net synthesize glucose sustainably. Instead, you would have to have other anaplerotic reactions that were present in order to keep that going. Glucose is not the only thing we can make from the pathway, however. We can actually make amino acids from components of the pathway, similarly to how we could take amino acids and make components in the pathway. The key things for now are that basically the citric acid cycle is going to be very important for a variety of different things, and it serves as a sort of central hub. If we use it as a cycle and just keep feeding in acetyl-CoA without taking anything out, then we're going to get we're going to get a bunch of NADH, a bunch of FADH₂, some GTP, and this is going to give us energy. But if we want, we can take things out of the pathway. And if we take things out of the pathway, then we can make other things. We can make, say, glucose. If we take out malate, because we can't take out oxaloacetate, but if we take out malate, we can use malate dehydrogenase to make oxaloacetate, and then carboxykinase to make phosphoryl pyruvate, which we could take through gluconeogenesis in order to make glucose. Or we could basically take out citrate. We can take out citrate to make fatty acids. We take out citrate rather than acetyl-CoA because we can't take acetyl-CoA out of the mitochondria. So citrate and malate, these are the two kind of points I want you to know. Citrate is taking out to make fatty acids, and malate is taking out to make oxaloacetate, which you can use to make glucose. So those are the key kind of 2 points that I really want you to know in terms of what we're taking out to make what. I also want you to know the regulation. So the key regulatory points of the citric acid cycle were going to be our pyruvate dehydrogenase, our citrate synthase, our isocitrate dehydrogenase, and our alpha-ketoglutarate dehydrogenase complex. These are going to be inhibited by signs of high energy like ATP and stimulated by signs of low energy like AMP and ADP. They're going to also, a lot of them, be inhibited by NADH because, well, if you have a lot of NADH already, you don't want to keep this pathway going. If you have a lot of energy already, you don't want to keep this pathway going as the cycle to break things down. However, we still might want to have isolated steps of this pathway happening. And we can do this because those isolated steps are going to be reversible. And sometimes we'll have like different versions of these reactions happening in our mitochondria and in our cytoplasm. So these enzymes can be doing a lot of different things other than working just as this cycle. But when we think of it acting just as this cycle, we're going to regulate these key regulatory points. These points where we're losing CO2, we're generating NADH, et cetera, et cetera, et cetera. These other parts of it, however, well, now we can use those pieces, use those enzymes, make various things. There's a lot of different things that we could do with the components of the citric acid cycle, rather than just use the cycle as a way to generate the maximum amount of energy. However, in order to keep this cycle going, we're going to need anaplerotic reactions where we're sticking something into the pathway that's greater than 2 carbons. We're sticking something into the pathway that was already in the pathway rather than just adding another acetyl-CoA, because we need a way to allow more acetyl-CoA to come in if we take things away. If we add something that was already in the cycle, then we're going to be okay. But if we just add acetyl-CoA and we take anything out of the cycle, we're not going to be okay. because everything in the cycle is either going to be 6 carbons or 4 carbons or 5 carbons, whereas acetyl-CoA is only going to be 2 carbons. And so if we take anything out, then we're depleting our cycle and we can't keep going. Thankfully, we've got those anaplerotic reactions that can keep the cycle going.